Good afternoon. Welcome to the monthly Global Health Forum. My name is Majid Azadi and from the School of Public Health. And together with Peter Burney and Nicolette, we have actually been sort of envisioning and coordinating these sessions, which have actually been quite interesting, all of them. And we do have another interesting one this time on the topic being water. And actually one of the things that makes all of these global health fora quite interesting is the diversity of speakers and topics. And this one is no exception. So a diverse and group of speakers looking at water from different angles. So we have 4 speakers. They'll each present for about 15 minutes, and maybe we'll have time for sort of one, or if everything is on time, 2 questions, just so that there can actually be time for discussion at the end. And again, the discussions at the end turn out to be quite interesting because different perspectives come on always interesting presentations. So the first presentation is by Dr. Alex Webb from Faculty of Medicine, who actually works on I guess proteomics for detecting schistosomiasis and its precursors. So Alex, please.
Hi everyone, my name's Alex. I'm a postdoc in Professor Paul Freemont's synthetic biology lab, and I'm a microbiologist. And one of my research interests is engineering bacteria to make whole cell biosensors. And in this case, we can talk about a whole cell biosensor we've engineered to detect for the parasite, the family of parasite Schistosoma, which I'll go into in a bit more detail just next. So, why are we interested in making biosensors for schistosomes? Well, it goes back to our first funding we got from the Bill and Melinda Gates Foundation. We wanted to make a cheap, rapid response and easy to use biosensor that people could use in the field. So if they want to go and test if their water's been cleared, or if the parasites have come back, is it still there or not? So that's the idea behind our work. But why do we want to look at schistosoma as an example? Well, 200 million people worldwide are infected at the moment with schistosomes. There's a further 780 million people at risk of infection. And the main areas are infected are like the sort of developing worlds, Africa, sub-Saharan Africa, the Arabian Peninsula, China, Indonesia, places like that. But interestingly, it's actually come back to Europe. So due to global warming, they've found a re-emergence of this parasite in Corsica. So it's not just the developing world; it's over in Europe as well at the moment. And also, this is quite a sad statistic as well, which makes it more into why we want to do this. Literally, 280,000 people die in sub-Saharan Africa alone from this parasite. Okay, and it's also a neglected tropical disease. So there's not much research being done on this. So that's why we're interested in it. And it's just a nasty cartoon of the actual 2 adult parasites that you have in you. So the male is the bigger one, the big red one, and the female is the small pinky one inside. Okay, it's not a nice parasite. But okay, so we need to know a bit about the parasite's life cycle before we can actually decide where we're going to target our biosensor to. And this just shows you the life cycle of the parasite. So the adults, this doesn't even work, does it? You can't see that, can you? Can you see that? Yeah, okay. We're chilling around. So basically the adult worms mate in the human host or any other mammals and also birds. And they actually can cross-infect. So there are some species that can infect cattle as well as humans, and cattle, rats and humans, goats, humans, those sort of things. So they mate inside the human host, in this example, and they produce eggs, and they come out through your urine or feces. A lot of the developing world, people bathe and use that water in the fresh water for eating as well. So you can imagine that the feces and urine will get into those water courses quite easily, the fresh water. The eggs hatch, and they go. The myricardium larva goes and infects the intermediate host, which is freshwater snails. And it's all different species of snails, depending on the species of parasite. So it's not that simple. They then mature in those snails. The snails can live for years, and they can release up to 10,000 parasites per day. They keep living for years and years. We've got a colleague who has a tank of infected snails. He's had them for God knows how long. So they do survive this process. So, and some people are thinking of maybe this could be the site to try and target to get rid of the parasites, but we've all seen what happens when you try and take something out of the ecosystem. It doesn't work. Then they release the eggs, and then you get the invasive larvae, the cercarial larvae. And this is the site we decided to target our biosensor to, because this is the invasive part of the parasite. We know that if we can target this, we know we've got a population that are present and actually able to infect people in that water. Okay, I'm saying we're going to target that larva. What specifically do we target from that parasite? Well, when the cercarial, when they're released in the fresh water, they swim towards the shallow areas where people will be more likely to be, or the cattle will more likely to be. And they're attracted to heat, they're attracted to the light to get to the shallow water. They're also attracted to fatty acids in your skin. And when the larva makes contact with the skin, the head and tail separate, and the gland contents in the head release a cocktail of enzymes. And one of those enzymes is called elastase. The elastase breaks down your skin so that the head can get inside you. Okay, it's not like your skin will slough off. It's like small wounds so that they can get in through that layer. And we've, back in 2002, a group published a synthetic motif for this enzyme. So basically when enzymes make contact with proteins, they recognise amino acid sequence. And someone's recognised the sequence that elastase can bind to and cleave, in synthetic sequence. So we've taken that sequence and we've made a modular biosensor on the surface of bacteria. The nice idea of our biosensor is the modularity. So basically you have like an anchor which sits into the bacterial surface. We have a recognition motif. So we know what this enzyme recognises. We can put a small motif here and we can interchange this, and we've done that for loads of different things. And in this case we have a tag, so we can actually see if we've got cleavage. So basically, in this example of our biosensor, the bacteria are exposed to the enzyme. If the enzyme's present, it will cleave our motif and it releases the tag. And then we add like a colour compound which should bind the tag. If the tag's not there, then bacteria will be clear. If the enzyme doesn't recognise it, doesn't cleave the tag, we add our dye and we get a red pellet. Okay. So basically, what you want, if you've got the parasite in there, you'll have a clear pellet. If the parasite's not there, you'll have a red pellet of cells. This just briefly shows you the sort of workflow our published biosensor uses at the moment. So we have tested it on real live samples. It's not just a normal science paper where we haven't actually tested the parasite. From a colleague in Nottingham, we got some cercarial. We then cause them to separate, to turn into schistosoma, and so they release all their enzymes. We've then taken that liquid, lyophilized it, re-engineered it when it's come back to my lab, and we exposed our whole cells to this enzyme. And then you do this workflow here. We analyse it by adding our colour compound, and then we quantify it using a flow cytometer, so we know how much redness is there, or how little redness is there. And we also test it against commercial proteases. So I said we use two engineered bacteria. I didn't say we use two engineered bacteria. One is E. coli, and we use E. coli because it's rapid and easy to use in our lab. So I can make lots of different biosensors very quickly using E. coli. I can test them against a range of enzymes, see if they'll be cleaved by enzymes they shouldn't be cleaved by, or if they are recognised by the enzyme I want them to be recognised by. And also we use B. subtilis. Bacillus subtilis is a soil organism. You've probably heard of it before. It's generally regarded as safe by the FDA in America, and that's one of the reasons why we chose that as the biosensor we would use in the field. Because we said we wanted to make a rapid, easy use in the field, in the site, and it's recognised as safe by the FDA in America, so it should give us a good chance of being able to use this, get approval from European agencies, or the British agency, depending on what happens. Again, I just want to reiterate, we have tested our biosensors against real living biological samples, and we've got real repeats of the cercarial. This just shows you, this is actually the real data we've got. So basically, this shows you the E. coli biosensors. Okay, so along here, there's 3 different biosensors tested. So we've got MCPX-TEV, so that is a control biosensor, positive control. That recognises the tobacco etch virus. So there's a lot of published papers on tobacco etch virus. We know what motif it recognises, we know how quick it recognises stuff. And so that's the one in the clear box. We have our elastase biosensor in the middle of those 3. So that's the one designed for elastase. And then we have the control, or con. And that's where we've just taken the motif region and just put a bit of junk DNA in there. So a sequence that shouldn't be recognised by any enzymes. And then here we have 3 biological repeats. We had 3 separate samples of cercarial supplied to us. And you can see in all 3 of them, the elastase biosensor was always cleaved more than the others. Okay, and we know, and this is a real biological result. And we know this. There is different amounts of elastase, natural elastase, in those samples because we've tested them against a fluorogenic compound. So it's just that in those different samples, some of the cercarial release more elastase than the other ones. So that's how it is. And this is just the same experiment but with the bacillus biosensors. Again, okay, it only recognised one biological samples, but that's the biological sample that had the most natural elastase present in it. But here there wasn't as many off-target responses, so that's encouraging. So I said we want to use the bacillus biosensors in the field, but a lot of these places we're going to, so we've got colleagues who are already out in Africa doing field tests, looking for cercarial tests in water. A lot of them, there's not much cold storage, cold chain access, and one of our colleagues said their lab is basically a pickup truck in the desert. So it's like, you need to have a way of being able to transport your biosensors out there. So bacteria, they're not that good. You've got to keep them at certain temperatures, they can die, they can grow. You don't want to take them to places like that. So, but with bacillus, you can actually freeze dry them, and they're still viable. And this experiment just shows that. So I took 4 of my strains, so 3 encoding those 3 separate biosensors, especially the elastase one, and just an empty vector as a control. I freeze dried the bacteria. I then left them for a while, reconstituted them, and then we made them produce the biosensors again. And this just shows you that through the freeze drying process, the plasmid that encodes for the biosensors is maintained, and those bacteria that freeze dried can revive, can grow again, and make the biosensor, which is important because then we can take our little test tubes of the biosensors into the field, or a tray of them into the field in a freeze dried format, and then we can just reconstitute them with the water at the site. So in future, we obviously want to test, this is all lab based, we obviously want to test it in the field, and we've got different collaborations going on with different people in certain different countries. But the idea here would be that we would, it would come into this point here, so we'd have our biosensor, okay, the biosensor's there, we'd have our biosensor, and we'd expose it to different amounts of water. But as you've seen with the data I've shown you, you can see, hopefully you did see that if you have different amounts of elastase, the biosensors do behave in a different way. Yeah? You need a certain level of elastase there. And so one of the things we're developing is making a small trap so that we can encourage more of the schistosomes, if they're in that water, to come towards a local area or maybe into our trap. They'll go into the trap, they'll release their contents, and maybe we can just take that liquid and add it to our biosensors. The trap design is still we're still thinking of different designs for the biosensor trap. But the idea is we do realise we do need to have a way of getting more of the schistosomes together so that we can get enough to be able to analyse if it's there. But another thing, so this is something else we've just recently published. I know, so we're in our lab, it's synthetic biology, but we do a lot of responsible research and innovation. And so we do realise that some people don't like the idea of viable bacteria being released into the field for experiments. So one thing me and my colleague Richard just there, we've just developed a beefed up to this cell-free system. So basically cell-free is a transcription and translation system. So what you do is you take a cell and you smash it open, you sonicate the cells, and so you basically release all the good stuff inside, the transcription and translation machinery which transcribe and translate your DNA into proteins. And you add an energy mix, and you add your DNA. The DNA is the part that will encode your biosensor to make the protein of choice, i.e. whatever you want in that system. You add these 3 together, and then you just leave it incubating. So what we've shown is we can optimise protein production, and in this case the protein is just GFP, a green fluorescent protein, very simply. And that gives us the choice of being able to make just cell-free biosensors. So I could put a protein, or the DNA to encode a protein, like maybe split GFP, with the elastase motif in between the 2 halves. When it's cleaved by elastase, they'll be able to come together, and you can get green fluorescence protein, GFP, mature GFP produced. Or you could change, you can put whatever you want in there. We do lots of different things in cell-free. So, but we also need to think of the future. So the modularity of biosensors means that you can detect anything you want. So one of the interesting things is it could be that the elastase actually confers speciation as well on the different parasites. So we know that the different members of the schistosome family infect different hosts, and we know that they use elastase to get into these different hosts. Is there a chance that the different elastase work on different proteins, where they recognise different things? So we can easily test that if we can get the sequence of the different elastases. We can test them against a whole range of different biosensors and see if we can actually make biosensors that can tell us, okay, there's Schistosoma mansoni in there, there's also Schistosoma japonicum in there, there's also some hybrid schistosomes in there as well. That would be the overall goal, if that's doable. But the schistosomes also produce other proteases and peptidases, so we could always make other biosensors that can detect for those proteins, and for other parasites as well. And there's also other biosensors we can detect for. So there's some things called parasite-derived exosomes. So like lipid, like little vesicles that some parasites release, and they also have proteases on the surface. So there's nothing to stop us detecting them. An example there is Leishmania. So that is known to produce these as it's injected from the sandfly into the human. And also, some other groups do make DNA-based biosensors, so it doesn't have to be protein-based biosensors. But we do believe that the protein ones are going to be more easy to make. I think they're going to be more reliable because proteins don't really mutate or evolve that quickly, whereas DNA does change a lot. So if you're detecting a parasite based on its DNA, you can quite easily change its DNA you're targeted for. It's likely we could do quite well. Okay, so I've just got some people to thank. So obviously Paul, Richard's here, who I collaborated with, and Kirsten. Prof. Mike Duenhoff, he supplied the actual schistosome samples, and Joanna Webster, who used to be at the site at Imperial, she's also at the veterinary school, who helped us with the ideas about elastase and things like that. So that's that.
Perhaps time for one quick question, and there will be time for discussion at the end.
I'm sorry, I have a question about your cell-free
Yep.
procedure. When you do that, do you have something like a control group to?
Yeah, so the cell-free biosensors, yeah.
Yeah, you just take off the cell, the surface of the cell. I don't know how to say it in English. It's a different.
Yeah, so I only touched it briefly. So when you smash open a bacteria, you spin them down, and you get the transcription translation machinery. We do controls between batches. So basically, the data I showed you, the GFP, is actually the control we use. So we have an empty vector which doesn't encode for anything, and we measure this we take the cell-free reactions, we put them into a plate reader, and so we're looking for fluorescence. So the natural cell-free reaction does have a bit of a fluorescence. But you do two reactions. You have one which is just an empty vector, empty plasmid, so it shouldn't encode anything that you can measure. And the other one is the GFP, and so we can see, okay, we know that this batch we're making, I don't know, like we're getting this high level of GFP expression. But if we make another batch and it may be a bit less, there is batch variation with the cell-free. But obviously, yeah, we do that quite a bit, and there's other things you can do. So we can make, so we do have a whole library of plasmids where we change the promoter strength, so that we know that, okay, so we have like a wild type promoter which gives us X expression, but if we change the promoter we get less and less and less. So we do know that the cell-free is working because we're seeing what we expect. So if we engineer it to be not strong, and we put it in the RBS for that plasmid, for that GFP, if it's not as strong, when we see that in a result as well, when we know it's roughly working. It's not, there is batch variation with every cell-free. Whatever cell-free you make, even commercial ones, and a lot of time the commercial ones are not as good as the ones you can make in a lab anyway. So yeah, but that GFP is that GFP would be the control. So if I was to make a biosensor based on GFP, I'd have as a control a plasmid that encodes for just the full length GFP that would be green straight away, so you can compare the times and how it makes. Does that make sense?
Okay. Thanks again.
Okay, no, thank you.
Our next speaker is Simon Starker, who is from engineering and PhD student, talking about energy implications of wash.
Hello, good afternoon. My name is Simon de Sterke. I'm a PhD student at Imperial College London at the Department of Civil and Environmental Engineering. I'm the only PhD student among among 4 people, of which 3 are PhDs. So you can see that for the occasion I overdressed instead of underdressing. I'm a mechanical engineer, so I don't know that much about WASH. But my research is on the linkages between energy use and water use in cities, with London and Mumbai as a case study. So for this, I'm elaborating a bit on a part of my research, and I'll talk about that today. So the title is The Energy Implications of WASH, of Water, Sanitation and Hygiene in Mumbai. So you may ask, why talk about energy when talking about water, sanitation and hygiene? Well, it's because you might think WASH, I mean, these things are fundamental values, and it's a fundamental right for humans to have safe access to safe water, so it should be done at any cost. And yeah, in Mumbai underperforms in terms of water sanitation and hygiene, so this should be fixed at any cost. But justly because it has to be fixed at any cost, the implications are so important because the energy costs are inescapable. And why is this important in India? Because in India already electricity use is supply constrained. So Mumbai deals less with this. So in the island city, there is a 24-hour supply of electricity. There are almost never blackouts. In the suburban area, it's also like this. But in a lot of India, people only have access to electricity for 6 hours a day, and it's quite unpredictable. So if electricity use increases, this will increase problems with load shedding and so on. Also, in terms of climate change and greenhouse gas emissions, the carbon intensity of the electricity system in India is very high. It's at 900 kilograms on average of CO2 per kilowatt hour, which is double that of the UK. And then because of the linkages between water and energy use, there can also be secondary effects that I'll talk about in a bit. So the rest of the talk is divided roughly into 5 parts. First, I'll introduce the water energy nexus. Then I'll talk a bit about Mumbai in general. Then about water and sanitation in Mumbai. Then I'll talk about the energy use that's currently that's currently being used for in relation to water in Mumbai. And then I'll discuss the energy implications to achieve full access to safe and clean water. This is a diagram by the US Department of Energy, which hopefully will still exist in 4 months, but we're not really sure. And it's good because it illustrates the whole water energy nexus, where the water energy nexus is basically the set of interactions between the water systems and the energy systems. So on the right, there's a hydropower dam. So there, the head of water is used to generate electricity. Next to it is a mine. Water is also required for fuel extraction and for fuel processing. And then there's a thermal power plant, which needs cooling water. Some of that might be released to the environment again, but some of it is also evaporated, so is lost. And that is required to cool down the power cycle so that electricity is actually generated. On the other hand, the water system also requires a lot of energy for pumping it to the end users, for treatment of wastewater and of raw water. And then finally, there's the end users, which require both water and energy for their operation and livelihoods, and a lot of services require both. So water and energy are inextricably linked at the end use. This is just a slide listing what I just said, and in the interest of time, I should skip it. So let's now go to Mumbai. Mumbai has a very long history that I won't talk about now. It's a mega city on the Arabian Sea. It has 12.4 million people, and that's the population of the Greater Mumbai region. The boundary of the Greater Mumbai region is not physical, the city just goes on, and the larger encompassing entity is the Mumbai metropolitan region, which is home to more than 18 million people. 42% or higher of those people live in slums or slum type dwellings. This is an estimate. Some estimates are higher. Some estimates say 60%. It's really hard to tell, and it depends on the correctness of the census. Sewerage coverage is quite low. Only 40% of people have access to decent sewerage, and the average water use is, depending on the source, quite high or quite low. So this 90 liters per capita per day is from an end-use study where someone actually measured all the water used for the different purposes just by asking people how much they would use and measuring the volume in a bucket or in a measurement can. And then the higher estimate is by the the Municipal Corporation of Greater Mumbai. So this one is a bit higher. This is not really measured. It's just what they bill people on. People don't really complain because water is really cheap. And by having this number quite high, they can also imply that the leakage is quite low. But basically, these two numbers indicate that nobody really knows how much water is consumed and how much water is lost. There's no continuous water supply, so depending on the zone, depending on the time of year, unfortunately, water is only available under pressure for 2 hours to 6 hours per day, depending on timing schedules. And then, as I said, the leakage rate, or what they call non-revenue water, because some of it might also be stolen and not paid for, is 25 to 30%, but it could be higher. 30% is the leakage rate of London, for reference. This is a map of Mumbai, where the areas which are hatched vertically are the original islands. The horizontal hatching shows the land reclamation that was done before 1900, and the dotted areas are the land reclamation in the 20th century. So and all of this area is now heavily developed. So most of what Mumbai is now used to be sea. The lines show the drainage channels for stormwater and for sewage. And basically what this shows is that everything goes with minimal or no treatment into the sea, and some of it comes back. And so that's why Mumbai has a reputation of being a bit smelly when in the warmer periods of the year, and that's indeed the case. Also because most of it used to be sea, there are some bore wells, but the water is very saline, so it's not really usable for water supply augmentation. With this segue, I'll go into discussing water sanitation and hygiene, the situation in Mumbai now. So actually, all the water that goes into the Greater Mumbai area is treated at some point, but it's not safe anymore at the point of use because there are so many pathways for contamination in between. One reason, and one major reason, is infiltration into the unpressurized system, which is basically dry for 20 hours a day. There's also a lot of local storage in plastic vessels or in rooftop tanks, so those are extra opportunities for contamination and for bacterial growth. People do have household purifiers, but according to the National Sample Survey of 2011, that's only 3%. Now this might be a bit higher, but household reverse osmosis appliances are things that are generally only available to middle or upper class people because they're quite expensive. A lot of people use bottled water as a solution, and the origin of the water is a bit dubious at times, so that's not always safe. Some water is transported by truck, so again this water sits in a container for a while, which is also not necessarily clean or is not clean between uses. And then as I showed, the wastewater treatment is very basic, or there is no wastewater treatment. This is an image from a book called Pie Politics, and this shows the so-called in informal settlements, the spaghetti tubes. So basically the municipal corporation provides no waters to these dwellings, no individual water supply, so everyone has like a pipe into one connection down the road, and then it goes up to their house. So you can see it's a bit crooked. There are cracks in these pipes. Some of them are in the drainage ditch, so there's a lot of possibility for infiltration, and when there's floods, they're all submerged, so a lot of pathogens can get into the water. And this is a lot of information, but I just want to show the first line here. So this is from a field survey in the poor communities in Mumbai from 2002. So it's a bit old, but the situation is still not resolved. So the first line shows that in one area, at least 1 in 3 families surveyed had someone in the family that was sick. And of those people, up to 1/3 had an illness that was related to water. And this table 4 here shows that the annual cases per 1,000 people in some areas, of diarrhea in some areas is 614. So this means that, that's 614 cases of a water-related illness probably per 1,000 people in 1 year. So that's incredibly high. As I said, wastewater treatment is not very advanced, and this map shows this. So underneath, I put a band here to show the scale of Mumbai. The red line indicates the Greater Mumbai area. So this is the island city, and this is suburban Mumbai. And in the island city, basically all the 3 wastewater treatment plants in Kolaba, Worli, and Bandra are all, basically there's only screening and grit removal, and then it's pumped, or it just flows into the ocean, and some of it comes back. In the suburban area, the water plants do have, the treatment plants do have aerated lagoons, so there is some form of secondary treatment, but it's not very advanced. It's also discharged again into the environment, and there's one plant, the Malat plant, which just has no treatment. So this is a very serious health risk as well. Yeah. Moving into energy now, I made some estimates for the energy use of the water treatment and distribution. So this is upstream from the use in Mumbai. There are two major water treatment plants upstream. One is the Bhandup water treatment plant. It's been termed the largest in Asia. I don't know if this is still the case, but it processes 2,250 million liters per day of water, which is about the entire water consumption of London and more. And this plant consumes 140 megawatt hours of electricity per day. There's also the Panjarapur water treatment plant, which processes less, 1,365 milliliters per day, but it has it has an electricity consumption which is much higher because of the elevation, and that's 864 megawatt hours per day. And so the inferred combined energy intensity of these two plants is about 350 kilowatt hours per million liters, and this is comparable to London, which has a similar energy an energy intensity for water supply and treatment of about 500 kilowatt hours per million liters. As I said, some of the water is supplied in batches with trucks. So this is a truck delivering water to a condominium in the island city in Colaba. So here this pipe shows where the access to the underground sump, from where the water is then pumped up. What I want to show here is that there's also an energy consumption related, of course, to the transport of these trucks and to perhaps pumping to wherever they get the water from. But all sources indicate that this, in the big picture, this water consumption is quite small, and it's also difficult to estimate. But I just want to show it so that you're aware. In terms of wastewater treatment, I was able to get data for the water treatment plant in Worli, which is in the island city. It's called the Lovegrove Wastewater Treatment Facility. It has a pumping capacity of 2,080 milliliters per day, but this is only used, the entire capacity of 8 huge pumps is only used during the monsoon when a lot of water needs to be pumped out. Normally it's about 400 milliliters per day in the dry season. This plant only has primary treatment, so there are screens and then there's some grit removal, and the inferred energy intensity is quite low, so 0.04 kilowatt hours per cubic meter. And this picture shows the entrance to the marine outfall. There is a pumping house after this for if the tide is high and the water needs to be pumped out. So putting this all together, I was able to make an estimate for the residential electricity that's related to water use in Mumbai in 2011. So these 200 gigawatt hours are for the water treatment and supply. There's a small part, and this is an estimate, of in-house pumping, so to pump the water to the rooftop tanks, or also for booster pumps when the pressure in the system is too low. There's some water heating as well, which is low because because of the climate, water heating is only necessary for 2 or 3 months a year. And then there's a small slice of about 20 gigawatt hours in 2011 for the wastewater treatment. So this seems a lot. This is 450 gigawatt hours in 1 year, and this is about 1/14 of the total residential electricity consumption in Mumbai, to put in perspective. So it's considerable, but when compared on a per liter basis with London, it's very low. So as I said before, the energy intensity of the supply is similar, but in London, a lot of energy is used for water heating, and this is not the case in Mumbai. And then much more energy is used for wastewater treatment in London than in Mumbai. So if Mumbai wants to become, sorry? Yeah, I'll wrap up. Yeah. So if Mumbai wants to become a healthy city in terms of water use, it requires a continuous water supply, full sewerage coverage, rehabilitated slums with pressurized water for example through standpipes, and also secondary treatment of wastewater flows. This is just a qualitative assessment of the implications for energy use. Leakage will probably go up when the system is constantly pressurized, so this will increase the energy demand. Demand may go up or down depending on because if the system is continuously pressurized, people have access to more water, so they might use more, but then there's less incentive to store water for the next 2 days, which is then thrown away when there is water available the next day. Trucks would be eliminated, point of use treatment would not be necessary anymore, but there would be a lot of extra energy use from sewage treatment. And this is just a bit of a thought experiment. So if there would be advanced water treatment at a higher energy intensity of 0.4 kilowatt hours per cubic meter, if population increases, water use and leakage stay constant, then the total electricity use for the water system may increase from 431 to 888 gigawatt hours. So this is considerable, and then becomes 1/7 of the residential electricity use in 2011. This is my last slide, just wrapping up. Water-related disease is a serious problem in Mumbai. The water system needs to be transformed. As I've shown, the additional energy use is considerable and warrants more research, but also because the data are very uncertain and dependent on source. So it is important. We don't know exactly by how much, but it needs to be investigated. Thank you.
We'll save questions for the discussion just in the interest of time. So our next speaker is Pauline Schulbeck, who is returning from Imperial, who was here for years, and talking about salinity and hypertension.
Yeah, so my name is Pauline Schulbeck, and I currently work at the London School of Hygiene and Tropical Medicine, where I'm a research fellow. And I'm very interested in the effects of environmental change and climate change on population health, and in particularly if that goes through drinking water and nutrition of those populations. And the work that I'm presenting today was a study that we finished about a year ago, and was part of my PhD that I did here at Imperial as well, where we looked at climate change and drinking water, and especially the problems of salinity in drinking water, and the public health problems that this saline water brings along. So the study kind of it was quite anecdotal evidence that there was something going on in the coastal areas of Bangladesh and India. There had been a few studies, but our engagement with drinking water salinity and possible population health in Bangladesh actually came through a doctor that worked in the coastal area of Bangladesh, somewhere here, and he said, I have the feeling that just anecdotal evidence, but that there is many more women presenting with preeclampsia in my clinic than I'd seen before, and I'm not entirely sure, but I know well, preeclampsia is a disease that is in pregnant women that is characterized by high blood pressure, and he said I'm not entirely sure, but it might have something to do with the salty water that we are drinking here. We all know that dietary salt increases our blood pressure. There's a lot of studies done to that, but we weren't entirely sure if salt that was dissolved in water, if that would have the same effect. And he asked if we could help out. So my colleague Anira Khan did a study where she looked at preeclamptic women and healthy women and asked them to take a sample of their drinking water source, and indeed she found that the women that presented with preeclampsia were drinking on average from much higher salty drinking water sources than the healthy women. So we wanted to elaborate this study a little bit and see, well, if we see this in healthy women, surely this should also perhaps happen in the general population, with the difference that those people never get their blood pressure checked, because high blood pressure usually goes without symptoms unless you obviously become ill from it. And those women were at the clinic because they were going there anyway for their antenatal checkups. And also we were interested in, so how does that exactly work? I can imagine that if there is a flood and people are drinking from an open water pond, that the seawater goes in, but once they have then emptied the pond and wait for the rain to come back in, why is it still very, very salty, and where does that salt come from? So it was not only the epidemiology department that was involved in the study, but also the civil and environmental engineering department at South Kensington, who especially looked at that question of where does the salt come from, whereas we looked at once you drink salty water, what exactly is the effect on blood pressure. Quite an explorative study. We can't really tell you exactly the effect of drinking water salinity on blood pressure and number of strokes, but at least we got quite a good indication of what kind of magnitude we are talking about. This is the Bay of Bengal, and why is Bangladesh now specifically very vulnerable to those salinity problems? Well, that's first of all because it's an area where there's a lot of tropical cyclones, which are followed by storm surges. The area is very low-lying. You see that the purple ones is 1 to 3 meters above sea level. Once there is a flood that obviously reaches like quite far inland. The geomorphology means that the bay is horseshoe shaped, and I've been explained that also increases the wave height if a wave attacks the land. It's very very densely populated, so you can imagine that also brings along more problems and an extended population affected by these salinity problems. And then specifically for Bangladesh and a little bit less in India, there's a lot of problems with arsenic. So if people say, well, okay, we leave those ponds, they're very salty, let's drill a deep hole and start drinking from deeper layers. Not in the area where we studied, but there's quite a few areas where people have to deal with those problems of arsenic, and we obviously don't want to solve one problem but cause the next health problem. And then there is a few man-made issues. There was a dam built in India which made the flow in the river decrease. So if seawater comes up, there's less of a pushback from the rivers. And people were engaged in shrimp farming, which sometimes meant that there were some embankments in which there was only fresh water. But for the shrimp farming, shrimps grow better in brackish water. They sometimes would manually break the embankment and let the seawater in. So this would only exaggerate the problem. And of course, what we hypothesized was that climate change was very much increasing these problems, as it is related with an increased frequency of tropical cyclones, for example, that were hitting Bangladesh. So this was from a slightly older study, or at least I took the data from that, where they measured salinity in tube wells. It says tube wells, but it's shallow ones, so still there is quite a lot of seawater seeping in and out those kind of higher aquifers. And then you see indeed that especially in this area, the salinity levels were extremely high. And later in the study that we actually did, we found a median sodium concentration of 800 milligrams per litre. And just to put that a bit into perspective, there's not really a WHO, World Health Organization guideline on how much salt you should maximum have in drinking water, but they say, well, let's aim for about 20 milligrams per litre, and the FAO is talking in their reports about a taste threshold. So they say, well, if it's above this, people would probably not really like it anymore. So we presume that no one drinks water, and that guideline is 200 milligrams per litre. So you can imagine we're talking, nobody really expected that there would ever be people drinking this high level of sodium in their drinking water. And I really hope that the WHO will also pick this up and develop a guideline, so at least governments could work with that. And it was actually really salty. I've tried to drink it a few times, but I could physically not swallow the water. But I guess because they got gradually kind of used to it, for them it was a bit easier to drink. Plus there weren't really any alternatives, so they had to go with it. So yeah. So the main question was like, so does this affect the blood pressure the same as all those studies that have been performed on dietary salt and water? Well, the first two slides I'd like to present come from the Civil and Environmental Engineering Department, so if I get it wrong, please correct me. But this was, they logged for a very long time the sodium concentration in one of the ponds. They had a very big monitoring station in it, and we were kind of hoping is probably not a good word, because we obviously didn't want a flood, but we said, oh, if there is a flood, we can actually measure what is happening to the drinking water and how quickly it becomes more saline, etc., etc. The flood didn't happen, but what happened was that the embankment broke, which was kind of the same effect as a flood, and you see immediately the sodium concentrations going up massively, and around this point, I believe people stopped drinking the water, and a few months later they emptied the pond to wait for the rainwater to fill it up again. But what they found, and what they didn't really expect, well, this is the level of water in the pond, and of course we know that it fills up with rainwater, that evaporation is going out, and that people abstract water because they were obviously collecting water for their households, and we expected that there was some seepage to the groundwater layers, but what we didn't really expect was there was quite a lot of diffusion back into the pond from the very salty aquifer, especially after a flood, and so they also found that it very much depended on how quickly you took action to empty the pond and let it refill again with rainwater, how salty the water would be in the pond. And this is a model that Adrian Butler and his team are still working on. Oh, sorry, and there was obviously the storm surge water if there was an inundation. But these were the two questions that I was trying to answer. So how does the saline drinking water affect the blood pressure? And what is the prevention potential? If you, for example, would provide people with rainwater-based systems, and how would that affect their blood pressure and their events like strokes and heart attacks, etc., etc., if we would put that in place? Spoiler, that last part I didn't really get answered, but the blood pressure one I did. So we were lucky because there wasn't really enough money to really put in a complete new intervention study, but UNICEF and the University of Dhaka were building a few so-called managed aquifer recharge systems. So with systems they captured rainwater, and they, with the gravity system, they stored that rainwater in the ground during the rainy season, so that people could, and it sat there as a fresh water bubble. There wasn't any cement or so around it, and and then people could bump it up in the dry season, and then obviously this bubble would become smaller and smaller and smaller, and it would start mixing a little bit with the more saline water around it. So towards the end of the dry season, it wasn't very high quality, but at the beginning, you had relatively fresh water and a good alternative for the pond water. There were also some solar distillations initiatives. They were much smaller, and there was some rain water harvesting. But we especially were interested in these systems. So we selected quite a few households we expected more or less half to start using this system halfway in the study. And so we followed the people up in terms of the drinking water salinity and their blood pressure, and then see if once they started using less saline drinking water sources, that we would also see a dip in their blood pressure. So this is a bit the data that, or the study design. So we looked at the drinking water salinity of all the drinking water sources that people were reporting, the cooking water sources, and we collected urinary samples to look at sodium in their urine. Then, of course, we had to measure everyone's blood pressure, systolic and diastolic, and then there were a very high number of confounders. We obviously needed to adjust for salt that they were eating with their food, because that ranged quite a bit as well. And then there is other things such as physical activity, BMI, smoking, age, sex, of course, and a number of other confounders. So the baseline data were collected in March 2013. The MAR systems started to become operational here in January, and then we measured, there was actually also one in April, but we measured them a few times after the MAR system was into place. And also this is quite relatively in the middle of the dry season, and this is really towards the end of the dry season. So people that would not, people that would drink from the MAR system, slowly they would see an increase in their salinity again. So we could compare between years and within like kind of a dry season if we could see that link between drinking water salinity and blood pressure. So there were a lot of results, and I picked this one where I grouped the people into substantial increase between time points in their drinking water salinity, substantial decrease, or more or less the same, because I believe that that's the best way to explain it. And then those two are from the between year comparison, the March and March, and those two are from the within year, so over the course of the dry season, the beginning of the dry season, compared with the end of the dry season. And then this is the systolic blood pressure results, and this is the diastolic blood pressure results. So what you can see is that compared to the people that had no change and just continued drinking water of the same concentration of salinity, those with a decrease in their sodium experienced a significant decrease in their blood pressure as well, and those with an increase in their drinking water salinity, a significant increase as well. And also, I looked at the per 100 milligram per liter step increase in drinking water salinity, and that was associated with 0.95 over 0.57 millimeters mercury. And if you remember that at the beginning I said that the median drinking water sodium concentration was 800 milligrams a liter, you can imagine that if you would provide someone who's drinking from a source that is 800 milligrams a liter, and you would replace that by rainwater, you have to multiply this by 8. So that's quite a substantial decrease in the blood pressure. Also, the odds of hypertension were 14% lower per each 100 milligrams per liter drinking water sodium reduction. The only problem we found with MAR systems is that people reported diarrhea more frequently than as compared to, for example, tube wells. Which was a bit of a problem. The MAR systems are very much under construction still, so people would are very much in development. So they are working on making it more of closed systems, and hopefully then the diarrhea will go down as well. So these were, I think, so although it was a very explorative study and we included about 600 people in this study, I think it was quite a nice way of first having that link, or having some evidence for that link between drinking water salinity, problems that might increase in the future due to man-made issues, due to climate change, and blood pressure. And because there's a lot of people living, in Bangladesh alone already 25 million people living in those coastal areas, and then India and Myanmar, etc., have the same types of problems. So these problems will increase, and we have to take this very seriously. So I think in this research we managed to at least find some supporting evidence for that hypothesis that indeed we do have a population health problem here, and that it could be reversed. That if we do something about it, it's a very reversible problem. And I hope also that from a policy point of view, that these maximum guidelines for drinking water will be there, because that's something that governments can work with. And then I think the MAR, although there were quite a few problems with it, is one of the possibilities that we can explore as an alternative. So then the last slide. This is the second part, which I can't give you any results on, but we're working on a mathematical model to see. So with the with the salinity intake and the differences that you see over the seasons, what kind of number of strokes, myocardial infarctions, and stroke and myocardial infarction death can we prevent if we would provide people with another system, with an MAR system or a rainwater-based system? And so how much would it cost per life year lost, for example, or DALI averted? And these are the ones that alter with sodium intake. So these are the people I worked with, and who did an amazing amount of work as well. And I want to end with my favorite picture from Bangladesh. This is the area where the study was conducted in the early morning.